table salt. Table salt, the salt used at home, so it's not that a big deal. Table salt. Then you have to be with a detergent or liquid soap. You need to have a liquid soap, so it's supposed to be there. Then you have to be with water. You're supposed to have water for you to do this experiment. And then you need to have some plastic bags. Then you need to have what you call ethanol, alcohol. Yes, ethanol, alcohol. So you need to have it so that it's also helping us in the extraction. Then you need to have test tubes. If you don't have test tube, you need to have maybe something like a syringe. Yes. So number one, what you need to do is to peel off this banana. Yeah, let's peel it off. Yeah, it's not for eating. Some people that will be saying yummy yummy. No yummy yummy here. Yes. So we put the banana inside the plastic. Put the banana inside this plastic. So it depends on how many students you have who are going to be. So that's my banana. That's number one. Number two, you need to mix this with this and salt. So you need to measure. You have to be able to measure a specific concentration. So it will be guiding you what you're supposed to do depending on the experiment you want to do. But if you don't have how do you measure it? If you don't have, so if I don't have, what do I do? Yeah, you can just estimate. Yes, you can just estimate, so that, yeah, maybe depends on how much do you need. Yeah, maybe that one is enough. Yeah, you can estimate. Then you put salt, but please follow the instruction based on this is just a simple guide. Yes, so show you that it's not that a difficult thing. Yes, so I'm just trying to estimate. Yeah, it's enough. Then now I have to add this detergent or soap. The liquid soap from home, you know it. Think, why do we need to add the liquid soap? Why do we need to add the salt? Why do we need to add the water there? Yes, we have to see each and everything. So the color doesn't matter. You need to keep the temperature below. If you don't have a thermometer, just work with it. So this is a stirring rod. If you don't have, you can use a spoon. So now I'm trying to stir. So after that, so that is our mixture we have made. Now what we need to do is to squash this. We can squash when the liquid is in there already, or we can just first squash it a bit. Why do we squash it? Basically, it's not a big deal, just to increase the surface area for the reaction to take place. You know that. I'm done with putting. So I'm just doing this so that I prevent the leakages from what? From happening. So now after that, I need to add what I've prepared here, this mixture into this. Yes, have to add that into that. So I pour. What am I doing? I'm extracting DNA from a banana. Yes, I mean banana. So now I can mix it properly. Now the next thing is to add alcohol or ethanol. In this case, when I add ethanol, I need to. So do I add it immediately? What do I do? This ethanol needs to be cooled down. Yes, this, please do not drink this ethanol, because this ethanol is how much? Is 90 what? 95%. Yes. So now what I do, I'm going to make this ethanol to go into this syringe. So what do? I'm gonna put this syringe, yes, in the fridge for 10 to 15 minutes just to cool it down. Yeah. So now I'm going to put it in the, so you see it's simple. You don't need to have so many material to do these extractions. So now let's go to the fridge and we put this. So now we're gonna put it in the fridge. So you need a sieve. You need a sieve which you are going to use to filter out this. So let's go to filter it out. So concentration. Now we need to add the alcohol or the ethanol in here. And then we can wait a bit as it precipitates. So basically that is DNA. The extraction is scoop it out with a spoon, and then when you view it under microscope, you'll see entangled structures. They can ask you, first of all, they can ask you to write a full report of what you have done. Number two, maybe a teacher can be, or an instructor can be observing what you are doing and then is not saying anything. So it means that you're supposed to be doing the steps correctly or the methodology correctly, and then maybe can be awarding marks depending on the skill is looking for. And then maybe they can ask you some questions. What is the purpose of pulping the banana flesh? Explain the purpose of the following ingredients in the extraction process: table salt or salt, detergent or dishwash, liquid soap, whatever you used, alcohol. What is the purpose of alcohol? What are the observable threads or clamped made up of? That thing we saw at last, what is it made up of? What are some of the experimental errors? Yeah, they can also ask you the experimental errors, and then what is your conclusion in this regard? So if you are to write a report, don't forget to write an investigative question. And then the aim, don't forget about the aim, to investigate whether the process can be performed to extract DNA threads from a banana, because that's what we are using, depending on what you are using. Suggested maybe safety precautions. You have to talk about some of the precautions you have used so that there is safety. Maybe wearing goggles. You know that alcohol is highly flammable, so you avoid direct heating of alcohol, yes, or bringing next to the flame. What are some of the observations you saw? Yeah, you can talk about here we are talking about with the naked eyes. DNA is visible as clump of white, maybe something which is white, sticky, or wet cotton-like mass, something like that. So it depends on what exactly you have seen, but that's what we have seen. Then you can talk about the discussion and the interpretation of the results. Pulping the banana flesh is basically that pulping is necessary for breaking of the cells or opening them, and also increase the surface area for the extraction. Then table salt or salt, we can say that it keeps the cellular proteins dissolved in aqueous solution so that they do not precipitate in alcohol along with DNA. So basically it's just to help us DNA not dissolving with alcohol so that they are separated. Then detergent helps us to dissolve the phospholipids in the cell and the nuclear membrane, which break down. When it breaks down the nuclear membrane, the cell membrane, this facilitates the extraction of DNA from the nucleus and the cell. Yeah, so it is better. It makes the process faster. Yes, of DNA extraction. Then we can talk about the alcohol. What is the alcohol used for? Basically, alcohol is used to precipitate the DNA from the solution. Yeah, so that now once we add alcohol, you will see that two layers are formed. The white mist or wet cotton-like mass will be formed, yeah, on top of the beaker. Yes, if you're using a beaker, if you're using a glass, whatever you are using, yeah, you'll be able to see those two layers. But after adding alcohol, so alcohol is very important in precipitating the DNA out of the solution you have, or the mixture you have prepared. Then you need to talk about the experimental errors. Definitely, maybe you can talk about temperature. Yeah, then you can talk about the conclusion. You have to talk about the conclusion, maybe a simple process using the apparatus, whatever you have used, can be used to extract DNA from DNA threads from a banana or from banana cells, because you have used banana. Whatever you have used still is fine. So something like that. So the practical of extraction of DNA is not that complex. It's just that people, when they hear about DNA extraction, they think that it's a very complicated thing. So that is it. It has been M.Saidi. We are coming back with many practicals and many experiments, so that these experiments, you can use them to understand the concepts of your content. Thank you very much for tuning. Don't forget to subscribe, like, and share for our daily